Hi, and welcome to this PTI webinar, GMP for Cell Therapies, What You Need to Know, with Nufar Gross and Imbar Bazili. The webinar will last approximately 20 minutes with time for questions at the end. If you do have some questions, please ask these in the chat box. I hope you enjoy this webinar. I'll now pass you over to Nufar and Imbar.
Hey everyone, my name is Imbar Bazili. I'm the QA director at biotech. I have 11 years of experience in the cell therapy industry. In my previous roles, I was the production manager in the cell therapy company for 3.5 years. Here with me is Nufar Gross.
Hi everybody. So I'm Nufar. I'm the general manager of biotech, which is an Israeli-based company, provide process development services. I've been in the company since it was established 3 years ago. And in my current position, among others, I'm the one responsible for GMP services like training and courses like the one we do today. I have more than 10 years of experience both in development and GMP manufacturing. And firstly, I want to introduce you to Bio, which is a global cell therapy innovation center, provides end-to-end solution for cell and gene therapies like scale-up, automation, process engineering, and additional GMP services. And we see ourselves as a bridge between science and technology in order to enable these great cures into the patient which need them so much. So today we will gonna talk about some of the things we need to consider while developing a cell therapy. First of all, I want to a little bit point out some of the differences between traditional pharma and cell therapy products. First of all, the active ingredient in traditional pharma is a chemical one. And in cell therapy, the active ingredient is the cells which are actually alive. And that means we cannot filter or sterilize the product at the end of the process. In order to make sure that we are not jeopardizing the patient, we need to work in a aseptic manner, which means if you imagine a regular cell therapy process, it means a lot of open manipulation, because when we passage the cells between flask to flask, we use to open caps of flask, bottles, tubes, and handling liquid between them via pipetting, which is very open manipulation and prone to contamination, and in very small movements it can cause turbulence just next to the product, which will eventually cause contamination. So in order to prevent that, we are using multi-level protection of both direct and indirect solution. What do I mean by direct and indirect? Indirect is the barriers that we have in the clean room, like the hair and handling system, like the hood, which allow us more clean environment to work in, sterile gloving, sterile gowning. All of those help us to contain the contamination that we bring with ourselves into the clean room and can jeopardize the product. In direct level, we mean we want to close the system as much as possible through the process, and by elements of sterile connectors, or use a welding for connect tube to tube, or even in a simple manner, just instead of using a lot of small volume vessels, going into one large single vessel. This will also reduce the amount of opening that we currently have in our process. So during our work in the GMP environment, we want to make sure that we are producing a safe product, and we are using environmental control to do that and to monitor that, as well as safety tests like sterility and mycoplasma during the process and at the end at the fill and finish point. Something that is extraordinary is treatment that are given as fresh cells, because they in those cases we have to do rapid sterility additionally to the regular sterility test that we do, in order to make sure that the patient has the most minimal risk that we can allow. Additional difference is that in traditional pharma, usually the process is very short, a few days, and is manufactured by machines in an automated way. But cell therapies are very complex processes which take a long time and is heavily dependent on manual performance. And sorry, and when we go to customer, we see that a lot of time they have one technician or two that is very experienced. They say that in their hands we all get the best cells, but that means for us that it's more than an art, than a process. And this is something when we are developing a cure, we want to avoid. We want that each time we enter the clean room, we will produce the same product each and every time. And a solution for that can be automation, although in cell therapy, usually automation is implemented into the process in a quite late stages, like phase 2 and 3, but we should start thinking on that moving forward since the preclinical trials, because we want to start think about a method that will be scalable through all the clinical stages. So in order to help us control and gain some control in the process, we use both risk management and process qualification, which you probably know as aseptic process simulation or media fill. The regulation wants us to prove that we are in control of our process, and we do that by media fill every 6 months for every one of our processes that we are manufacturing. To do that, we need to make sure that our employees are well trained and undergoes requalification by themselves. So we need to qualify both the process and the employees, and it's not a very easy task to do, because what we see is usually companies are more focused on manufacturing. They have few skilled employees, and when they get new hires, it's very difficult to find the resources to now train them. So it makes them more focused on training, on theoretical training rather than practical one, which are simulations and moving into the clean room and learning by the hand, because new hires cannot do clinical batches. So in this case, it's a great challenge, and some companies struggle a lot with that. Additional difference is the volume of the batch. Usually in traditional pharma, we talk about thousands of pills, while in cell therapy, we have, in the best case, a few hundreds, but we also have autologous treatment, which each batch is a single dose for one patient. This is very hard to manage and very hard for us to keep the production and stand with the demand. So for that, we need in allogeneic treatment to scale up, or in autologous treatment to scale out. No matter which one, we should think about it early on the development phase. Again, we want to make sure that each method we choose is scalable and will be implemented through all of the stages. Additional difference is the shelf life. In cell therapy, the shelf life is very short, which drives us to try and make deep freeze for the product, use cryopreservation. Cryopreservation for now is a process that can be done for cells, but impose by itself a great risk, because when we want to ship our product in liquid nitrogen to the hospitals or to the patients, we still doesn't have very good solution for that. There are a lot of work being done these days to make sure that we can provide couriers that with liquid nitrogen to reach everywhere, but it's still ongoing work. Additional challenge in cell therapy is the batch-to-batch variability. And there are a few elements that contribute to that. First of all, the variability between donors. Each donor has its own genetics, its own biology, and it can be variable very much between one patient to another. Adding to that is the differences in raw material. We have raw material like serums, antibodies, growth factor that they have a lot of inconsistency inside between their batches. So this is also something contribute to the variability. And finally, we have the personnel that deal with the packaging and the process, which can also increase the variability. So in order to make a consistent process which will be repeatable, we need to move forward for automation, or if we still have open manipulation, again, this is something that rely heavily on training, good training, making sure that all the employee is trained the same. And sorry, another point we should consider is that we need to collect data through all of the years of the development and manufacturing, because we want to know ourselves and make sure that when we switch to automation, we will know how to handle the algorithms and when to pump in media, when to take it out, and which is really the limiting factor at each point.
So there are some things you need to consider when designing the process for the clean rooms. Numerous times we've seen a process that was designed in the lab, but when reaching to the production floor, things take the wrong way just because the GMP constraints were ignored in the prior stage of the development. Some examples are process duration. When working in R&D lab, things are being done and completed very fast. In the clean rooms, where each activity comes with documentation and cleaning, it takes much more time. And in our case, time equal quality, because cells can start dying or even adhere to the container walls in some cases, which can affect the process yield and can cause a gap between the development and the production process. Another example is in R&D setting. In R&D setting, when operator needs to enter something into the wood, he takes his hands out, bring the missing item, spray the item and his hands with the ethanol and go back to the wood. In the clean rooms, each item should be both sprayed and wiped separately, introduced into the woods only when all the vessels are closed. The operator usually required to change sterile gloves or at least spray his hands and wait for the ethanol to evaporate before continue working, which means the steps are longer in the clean room. And this is only a small example. Overall, we need to figure out ways to simplify the process as much as possible. For example, do we really need to change media every other day? What is an obvious manipulation in the lab, in the clean room, it will require additional steps, and it's something that we need to take in consideration. Steps like clean room cleaning before and after the activity, room clearance, environmental monitoring, and other supportive activities that may not be related directly to the product, but can definitely affect the time and money required for manufacturing it. Additionally, manipulation requires high skill, are prone to human error, so it's a good advice to minimize it as much as possible. When designing the process, we always need to remind ourselves to look in a GMP process point of view. For instance, if we used to freeze a small amount of ice at the end of the process in the lab, when handling a batch in full size, it needs to go through a visual inspection and labeling while with the MSO, element that we tend to regard in the lab. It can cause us loss of cells and impact the yield and quality of the product. When performing the activity in the lab, usually the activity will be performed by one employee, maybe two. While in the clean rooms, all the activities needs to be performed at least by 2 people, and sometimes even more. This should be considered in the amount of trained personnel. Adding to that the fact that not all the employees are present in 100% of the week. It should be also considered when building the production team. As mentioned before, qualifying production personnel is not easy. So, and except of aseptic work in the wood, they should be also trained on other subjects like proper gowning, equipment handling, and other issues to support the manufacturing. So, why the training is so important? The decisions made in the clean rooms are based on experience and knowledge and can directly affect the product. For example, conference assessment will determine when to perform a passage. Making this decision can affect the yield if we harvested the cell at the wrong time. The aseptic techniques that will be used in the process can also impact the yield if performed in a wrong way, harming the consistency between one batch to another. Raw materials are also a great example for how GMP affects our daily operation. Another example, if you are used pipettes, which I believe you do, you can't order any kind of pipette that you want. You need to understand that also the quality of the package can affect the risk for contamination. In clean rooms, don't use pipettes that has paper side in the wrap and prefer ones that are individually packaged. About expertise, usually the knowledge transferred to a new team member is something that is more trainer dependent, but it's the little things that and attention that will make the difference and allow us producing a high quality product. When the training and qualification process is performed by the same team all the time with experienced people, the details won't be missed and you get an aseptic techniques robustness and consistency. Training of new hires is not the end of learning in GMP environment. In order to maintain the GMP standards, we need to perform refresh training on SOPs that were not trained in the last year to see the relevance of the instructions written in the SOP. Is the SOP still applicable, or on a daily basis things are performed differently? Generate an annual refresh training program, learn from your own mistakes and explore your deviations. Other deviation that happens again and again, if so, it's time to change something. As we mentioned before, GMP is not for the production staff only. It should be reflected in different departments as well. And they all need to be trained on GMP issues in order to give a full support to the manufacturing teams.
So in order to summarize quickly what we talked about, we define the key differences between our industry and the traditional pharma, and we spoke about the things we should consider since the moment, from the moment that we start creating our process and designing our manufacturing. So this is the thing we should pay attention when moving forward in the clinical phases. So I want to thank you all for listening and for your time. And thank you, Oliver, and all the guys in PTI for the great collaboration. And we will now be happy to answer any questions you have.
Okay, thank you both for that. So yeah, we just allow another minute or so for questions to come in if there are any, and if they're not, then we will probably finish. Okay, we've had our first question, and that is, are there rules for raw materials for cell therapies?
So first of all, any raw materials that we are using, we need to do risk assessment regarding that, which means we need to understand in which step we use. There are some regulation around using antibiotics, for example, and of course, the main raw material that need to be considered is if we want to use serum or not. And if so, when it's time to remove it from the process and how we can control the residue of it at the end of the process. So it's really depend on the raw material. And I will slightly extend the question about consumable as well, because in biotech, we need to make sure that we handle the items properly and the flow of the items from the warehouse into the clean room is appropriate, and we reduce a wrappage or a package each time we move between ISO, let's say from ISO 8 to ISO 7, we need to remove a wrap and an additional wrap when we enter the room. So that means we need to consider items that are multiple wrapped.
Okay, thank you. We've got a couple more. So we talked about clean room use. However, this is very expensive and may bring cost of manufacturing up a lot. What about switching to isolators in Class C or D background environments?
Okay, this is very good question, because there are a lot of movement going into isolator. This is really process dependent. There are some processes that we can use isolators, but when handling very large equipment like cell stack or 40-layer cell factories, this is something that we cannot do right now in isolators, or we need to build very big ones. So again, it really depends on the process you have.
Okay, so thank you. Next question then is, have you compared head-to-head all the different GMP compliant technologies and platforms for ATMP cell therapies?
Sorry, can you repeat that again, Oliver?
Yeah, sure. So the question was, have you compared head-to-head all the different GMP compliant technologies and platforms for ATMP cell therapies?
So again, this is really process dependent, because some of the processes handle adherent cells, and we can use several platforms for that. But when dealing with suspension cell, we have completely different set of technologies that we can use. So again, it's very dependent, and each one of the platform has its own advantages and disadvantages. We can compare bioreactors, for example, but each bioreactor or each carrier that we use will act differently between cell to cell or process to process.
Okay, thank you. So we've got two more questions, and that is, what are the main issues cell therapy companies encounter in regards to GMP?
This is a huge question, because again, first of all, this is very expensive to operate a clean room, and not only build one, but to make sure that all the equipment is validated, all the people is trained, do the qualification of both processes and employees. So all of it is very costly. So this is something not every company could afford. Some of them are going to CMOs because of that. What we want to to emphasize is that the training of the employees should be hands-on. So a lot of time when companies dealing with the fact that they need to go enter GMP, they forget they need to train those personnel, and it's very, very complex training. So this is something that they deal a lot.
Okay, so last question then, and the question is, coming back to isolators, if we imagine a process where only a few steps are done inside the isolator, can we then do many steps outside by aseptically joining tubing and equipment, for example, using tube welders?
If you are using welding machines, so you don't need the isolator. You perform welding in you can perform welding in ISO 7. So if you already bought the welding machine, it's better that you will do it in ISO 7, and even you can do it in ISO 8. So because it we refer it as a closed system. The isolator will be in ISO 5, so it's too high and too expensive.